All right. Hello, everybody. When I was asked to do a Newman talk, they gave me a bunch of different dates I could choose from, and I saw Halloween was one of them. And I thought it would be just perfect to do a recap of Mad Science. So I actually thought we should start with old Mad Science and talk a tiny bit about Frankenstein. Does anybody know what the subtitle to the book Frankenstein is? Yes.
A modern Prometheus?
A modern Prometheus. Who was Prometheus?
Figure in Greek mythology who brought fire to human beings and got punished for it.
All right, but he did something before he brought fire to the humans. All right, so he made the first humans, at least according to some, Greek mythology has a diverse set of myths in it. But one of the myths is that Prometheus was the person who built the first humans out of clay. Life was then breathed into those humans, etc., etc. Professor Haynes is right that then some of the other myths talk about him then stealing fire back for the humans after Zeus took that away. Some people say it was in punishment for Prometheus playing other tricks on Zeus. What was his punishment? Anybody remember what Prometheus' punishment is? Yeah.
Didn't he get stuck up on something to have his eyes pecked out every day?
Not his eyes pecked out every day.
Liver.
His liver pecked out every day. So I bring this up because actually it's an interesting choice for the Greeks to have picked his liver to grow back, because your liver is one of your organs that you are great at regenerating. Right? Had his eyes been plucked out, they would have never regrown. But interestingly, if you have just a little bit of liver left over, that liver will eventually regrow and could then be pecked out again. So kind of an interesting choice there. So there's a little picture of Prometheus with his liver getting pecked out by a bird while he's chained to the rock. Now, the story of Frankenstein was set in a very interesting time as far as science was going. So we all know the story of Frankenstein, right? The mad scientist assembles life, and then the lightning bolts come, at least depending on which edition you're looking at, and shock life into this inanimate creature, which then becomes alive, and then there's all the consequences that come from that. The context for Frankenstein being written was a time when people were beginning to try to figure out what made life life. All right, so there were experiments going on. This one up here at the top by Galvani. He was trying to figure out what he called animal electricity. The electricity that he thought ran through organisms' bodies. And he did experiments showing that if he put conductive wires between nerves and muscles, that he would cause those muscles to twitch, which he thought was proof that these cells were creating electricity and that this electricity was built into them. Now, he was right about some parts, he was wrong about other parts. But these experiments led eventually to people actually trying to do this on humans. The most famous experiment of this was a prisoner, I'm now forgetting the guy's name, Charles Foster or something like that, who was executed at Newgate Prison in London. And that afternoon was taken to a nearby house where they conducted very similar experiments on him, hooked him wires and tried to sort of shock him back to life. And they report that his facial muscles twitched and his eye popped open and things like that. There were actually all kinds of things they put into place at the time. They were worried, what if we actually do bring him back to life? Do we have to execute him again? So these were serious issues. Now, of course, he did not actually come back to life, but that was sort of the context of Mary Shelley writing this book at a time when people were really beginning to think about what was life. Was life something that was merely the sum of a bunch of physical parts, or was life some kind of gift from God? And so a debate that probably to a certain extent still continues today. So what I thought I would do is I would provide some snippets of sort of where our mad science is at now. What are sort of the experiments that are pushing us right up against the very fronts of, if I can use the sort of the phrase playing God, right? And I've got a whole bunch of them that I put up there. I'm going to just sort of give a very short description of them. If there are things you want to talk about or consequences that come from it, if you want to talk more about some, less about others, that's fine. We'll see how far we get through them. All right? So the first one, if we're going to be talking about life, is going ahead and trying to create life. Where are we at in trying to create life? This gentleman over here on the right there, his name is Craig Venter. Anybody know Craig Venter or heard of him before?
Something to do with genetic research.
Yeah, so he was, many people consider him to be responsible for the Human Genome Project. What he was actually responsible for was the private portion of the Human Genome Project. The Human Genome Project was to be this government-funded thing where they were going to slowly work their way and assemble the entire human genome. And Craig Venter decided that he was going to start a company and he was going to beat the government to the punch. And came up with new technology which nobody thought was going to work, and he synthesized many of the first genomes, was clearly on path to synthesize the first human genome, and was basically talking about patenting as much material as he could to make that then his company's information, which he could then sell out. That's going to be how he's going to make money for his company. Now, eventually, he and the government reached a sort of a happy truce where the government would use his ideas, but the information would go into the public domain, and they were able to assemble the genome much faster than initially thought. All right, so this guy sort of has a history of going out and trying crazy things, but being successful at it. And what his new goal is trying to create life from its chemical components, right? And he's slowly working his way down to that by making what he calls the hypothetical minimum genome. So they're taking the smallest genomes that they know, which in this case are bacterial genomes, and they're basically going through and they're trying to cut out all of the junk from them. He's also doing some cool things, or maybe not cool, depending on how you think about it, like putting in his name in the genetic code and little quotes from literature about, the hubris of scientists, things like that. So they're editing these genomes and basically trying to create the smallest possible genome. Then they're actually synthesizing that genome on a machine, so not using a biological organism to copy the genome, but actually writing it as little pieces of chemical, and then inserting that into empty bacteria where they've removed all the genetic material and basically creating what they call their first synthetic lives. They're now on what they call SYN 3.0, the third variant of this. And they're down to a genome that has 473 genes in it. Just for the record, our genome has somewhere on the order of 25,000 genes. So they're down to less than 500. Kind of interestingly, for those of you who are biologists, you are all biology. For those of you who are biologists, one thing that's interesting is out of that 470 genes that they're using for this, they don't know the function of 150 of them. Right? So all that stuff that we tell you in introductory biology about like how cells work, we're lying to you. Right? We don't understand at least a third of what's required for basic life here. Right? But Venter has this idea that basically life is a DNA software system, that if he can figure out all that's necessary in this DNA code, that he's going to be able to create life. He's going to be able to do whatever he wants with it. Now, he has actually very admirable goals in theory in this. He would like to create life that has the ability to, for example, create petrochemicals so that we're not reliant on digging up old petrochemicals, that we can actually like make gasoline from other things. So he would like to sort of edit these genomes once he's figured out how to do it efficiently to make bacteria do better stuff than what they're doing now. He's got another quote there, we're moving from reading the genetic code to writing it, right? We spend a lot of time trying to figure out what genes are doing, and now he's basically saying we're at a point of wanting to start changing that. What do you guys think about this? Cool? Scary? A little of both? Mad science?
Mad God complex.
Mad God complex? Yeah, he's got no shortage of God complex. If you go look up quotes about Craig Venter, he is full of like proclaiming that he's about to win the Nobel Prize and that he should have religions named after him. You know, he's not a shy gentleman. We can say that. I mean, certainly this ties in with sort of that the questions that were going on in the late 18th century, early 19th century about whether life is merely the components that go into it or whether life is something different. One of Craig Venter's quotes is that at the very least what he has done is showed that God is not necessary for life because he assembled all of the components, stuck it together, and made this bacteria that can keep growing. So like I say, not a shy gentleman. All right, so on this idea of moving from reading the genetic code to writing it, all of these things are sort of headlines from the last 2 years, most of them actually from the last year. You may have heard about this new system, CRISPR-Cas. It's been in the news quite a bit. It was actually in the news just last month. NPR did an interview with a Swedish scientist who's using this to start editing human genes. The basic idea of this is that scientists have borrowed a bacterial protein that cuts DNA in certain areas, and then using the DNA's normal repair mechanism and sort of tweaking it, which allows you to go in and delete segments of a genome and then actually add new bits of DNA to that. All right? So this is commonly used right now in labs. In fact, I'm doing it right now in my own lab. So we're creating one-eyed fish right now, little cyclopic fish. So we're doing that because we're really interested in understanding what makes eyes form and why we have two evenly spaced eyes. So this is a very common research tool, but there's basically nothing that prevents it moving from a research tool to being used clinically. So the very first studies using this clinically are probably actually ongoing right now. They haven't fully announced it because everybody's a little cautious when you're at the very beginning of studies. But they have shown that they can, for example, edit bone marrow cells that from patients who were affected with sickle cell anemia and repair the defective gene in sickle cell anemia. So this is basically like a gene therapy kind of thing. And they've fixed that in mice, and the technique almost certainly is going to work perfectly well in humans. So it's almost certainly is currently underway. Several labs in China have said that they are preparing to do CRISPR editing of humans to repair diseases. And then, of course, there's this story over here of that Swedish scientist who is not doing this on like just bone marrow cells or some isolated cell that he wants to fix, but is actually planning to take normal human embryos and start editing the DNA of those, more or less just to see what they can do with that. How efficient is it going to be? Can they produce germline past things, perhaps? This is kind of some crazy ideas there. You can also see going to Jurassic World land, right? That people are using this to start moving DNA between species. So they have taken some woolly mammoth DNA and moved it into elephant skin cells. They've taken specifically the genes that code for woolly mammoth hair length and color and stuck it into elephant skin cells. So we're not quite at fully cloning mammoths from this, but that's like, again, not far away from what people are currently doing. Thoughts on this thing? Yeah.
I'm more interested in the clinical part.
Yeah.
For example, I mean, obviously you point to a genetically, a disease that has genetic origins.
Yep.
Anemia. But what about diseases that viruses come into us and
Great.
attack us? Can we fix our genes so that they can't attack us?
Absolutely. Well, it depends. It depends on the exact disease you're talking about. But for example, so AIDS is a really interesting one because there are people who are known to have a defective receptor that is necessary for the virus to enter the cell. And they found these as people who should, for all intents and purposes, have AIDS, but don't get it. And they found out that if you're lacking a certain cellular receptor, the virus can't interact with your cells, right? And so there are thoughts that one of the things that you could go in and do then is you could actually go and edit other humans' receptors to make them, in theory, immune from this virus.
Well, how about the common cold? That's one of the biggest causes of worker loss of productivity.
The common cold is tricky.
Because they mutate.
They mutate so much.
Yeah.
But who knows what's possible in the future? Who knows what's possible in the future? I mean, certainly with this, there are not limitations to what you can edit, at least as far as we know. You should be able to edit anything that's inside a genome. So if we can find out what the common cold is using and that it's using something reliably and that that's not needed for other important aspects of a cell, you should be able to edit it in ways that make the common cold less able to do it. I'm sure people are starting to look at it. I don't know specifically that. I do know for AIDS that people are thinking about that, but it is raising a question. I think people probably have less concern about like treating somebody with sickle cell anemia. Editing somebody's genes in order to protect them from a future disease seems maybe a little bit more questionable. Like we're now editing somebody who's normal and we're making their genes defective on the chance that they might get AIDS. So this starts to raise questions about like, well, why not then just say, well, my muscles are a little bit weak here, right? So like, why not just edit my genes so that I'm a little stronger, right? I could probably be a better athlete if I had stronger muscles, right? I'm just kind of a weakling, right? So what's the difference between normal and sort of required functions for things? It's going to push us in a lot of interesting directions. Yeah.
Also, do we know what the side effects are yet from using CRISPR-Cas in humans? Like, would you increase cancer risk or something like that?
You probably won't change cancer risk unless the gene that you're editing has something to do with cell cycle or things like that. In other organisms, it doesn't seem to be affecting that much. It's a very targeted approach. So that's one of the reasons people like it, is that it just goes in and it edits the gene that you're trying to fix. You do your little repair there, but it doesn't really screw with the rest of the genome. So it's better than other approaches people have thought of for gene therapy, where you kind of randomly insert repaired genes into the genome, but those often end up in bad places and do lead to problems. This seems to be much more targeted, so a little more focal. So like I say, in other organisms, we haven't seen effects. There's no reason to think humans are going to be all that different. Human cells don't seem to be any different. Yeah.
Okay, this is more of a science question from lack of understanding, but when they they edit the genes of a few cells and then that propagates through, I don't follow how it actually applies to the entire organism. Right.
So that's a really important point. So when I mentioned that they were trying to fix somebody with sickle cell anemia, right? So all of your blood cells come from your bone marrow, right? That's why they do bone marrow transplants for people who have leukemia or things like that, all right? So the idea there is that if you took out somebody's bone marrow who has sickle cell anemia, right? Instead of doing a transplant where they're going to have to be on immunosuppressant drugs because it's not their cells, right? If you took their own cells out of their bone marrow, you go through and you edit their stem cells that make all of their blood, right? Fix their stem cells so that their stem cells now don't make the bad protein that causes sickle cell. Stick those cells back in, right? You've sort of done a self-transplant. So those cells are now the cells from that patient. So you're not on immune suppressant drugs anymore, right? And you've repaired the protein that's causing the problem so that all of the progeny of those stem cells now have the repaired protein, okay? So that's in theory how you can do this for certain tissues, all right? Now, this here is a little different, right? This would be actually going through and doing that same type of repair process or editing process, but doing it in a 1-cell, 2-cell, 4-cell embryo. And now all of the progeny of those cells, which is basically the entire organism, are now edited. All right? And that's what we're doing with our fish. When we do this, we inject a 1-cell or a 2-cell fish embryo with some editing DNA that we're trying to target and the little fine details in there, and then we edit their genome, and then all of their cells are edited, plus all of their progeny, because we've affected the eggs and the sperm, because that's progeny from those original cells. So in that case, it would pass on. The bone marrow transplant, that patient would not pass on the edited gene, because it would only be in their blood system that has been fixed.
Yeah.
Does that distinction make sense there? Yeah.
So that human embryo study, how is that getting past the IRB?
Well, so IRBs don't care about human cells. They care about adult humans or child humans, but not necessarily human embryos.
Okay.
We're going to get to some other aspects of early human life in just a minute. So save all your questions about like how this is ethically possible. We'll get there. All right, cloning. So where are we on cloning? Just as a little bit of information about cloning for those of you who don't know, the idea of cloning, like you've heard of Dolly the sheep, right? You take some cells, right? Most commonly you take skin cells because they're really easy to get. For Dolly's case, they took some mammary epithelial cells. So you take some of these cells that you can grow up in a dish, you get one of those cells, and then you take an oocyte, an egg cell, from an unrelated person, sheep, whatever. And there's a variety of ways you can do this. You can either take the nucleus out of this guy and stick it into the egg that you've removed the nucleus from, or you do some kind of an electrofusion where you actually cause the cells to fuse together. This basically then turns that into an egg cell that thinks it's been fertilized because it has a normal set of DNA. This egg has only a half set of DNA, because it underwent the process of getting rid of, so you only have the mom set of chromosomes there, right? This thinks it has a full set of DNA, it divides, and it goes on. You can either use this to make embryonic-like stem cells, or you can go on to make Dolly the sheep. All right? Now, as far as we know, nobody has ever done this for humans. That's my daughter. That's not actually a cloned human. But we do know that people have gone this direction with human cloning. People have made human stem cell clones, or stem cells from cloned human embryos. They've actually been done from patients as old as 75. So they took a 75-year-old patient and used that cell to generate an embryo that they then isolated embryonic stem cells from and kept those growing in a lab. In theory, if we can do this, there's really nothing that stops this. It's exactly the same process. It's just whether or not you take the cells from this and grow them in a lab, or you take the cells from that and put it into a donor mother, into the uterus where it would then grow. Now, the chances of this are actually quite low. This turns out to be relatively easy to isolate cells and get them to grow. That's much easier. Dolly, they only got one sheep out of like 500 eggs that they tried to use. So it's rather challenging to get this to work, but it's just a low efficiency thing. There's nothing that seems to prohibit that. So that's kind of where we're at with human cloning right now. What's next? All right. Some of you may have seen some news sort of related to human cloning, and again, perhaps indicating just how close we are to actually being able to succeed at it. The birth of a baby with 3 parents' DNA. All right, so this was an interesting case. It was a couple who had tried to conceive for like a decade, and they only had one stillborn child and found that there were genetic abnormalities. But the genetic abnormalities were really interesting. The genetic abnormalities were in the mitochondria. So we all took high school biology, and you remember mitochondria are the powerhouses of the cell, right? I could draw the little picture you all saw there, but that's what we all learn, right? The mitochondria are the powerhouses. Now, what's interesting about those mitochondria is that they have a little bit of their own DNA. This small little circle of DNA that makes a fraction of the genes that are needed for the mitochondria to function, all right? There's some really cool aspects of that, but we're not really going to talk about that, except for the fact that this one family's issue was not with what we call genomic DNA, the nuclear DNA, but was with the mitochondrial DNA. So the mom had a mitochondrial disorder that didn't affect her, but when she passed on that disorder, it affected all of the offspring. Okay? And the reason for that is that from this picture before, when we have like a sperm and an egg, sperm contribute essentially no mitochondria. So all of your mitochondria are derived from your mom, all right? And if your mom has bad mitochondria, then you end up with bad mitochondria. And there's some complicated things where it actually starts to escalate when there's problems in this particular case. So basically what they did here is they took an unrelated donor's egg, removed the nucleus from that egg, stuck in a fertilized egg nucleus from the mom and the dad. So they fertilized an egg, put that nucleus from mom and dad into this third person's egg, right? And so now we have an egg that has nuclear DNA from mom and dad, but mitochondrial DNA from an unrelated third party who doesn't have this genetic disorder. All right? And it produced a perfectly viable young boy. Now you notice here it says banned technique. The actual stuff was done in Mexico because that was the only place they could find a clinic that was willing to let them do this. The researcher is a New York doctor. The patients were actually Jordanian. So this was a very small world kind of thing. They brought everybody together from all over the place to Mexico to get this to work. The technique is banned in some countries and not in other countries. This is when they could not get by U.S. regulators, so they went to Mexico to do it. But shows how successful you can be taking an oocyte, removing its nucleus, and sticking a new nucleus in, which is essentially the same thing as cloning. Right? So this is about the closest thing that we've come to cloning. In this case, it was not true cloning because they actually took it from a fertilized egg, but more or less right on the doorstep of that. Questions or comments or thoughts about the mad scientists here? Yeah.
So the mitochondria's DNA is discrete from the rest of the genome.
It is.
This necessarily wouldn't be a pathway for, say, a group of polygamists, like 10 parents, to meld their DNA into a child.
No, so your nucleus can really only have 2 copies of each chromosome, with a few exceptions of chromosomes where there's ways to deal with that. Okay. So the headline here is a little scarier than it turns out to be, right? The patient, as far as all of the somatic cells, all of the things that most of your cells are doing, all of that DNA is derived just from mom and dad. But almost all of the patient's mitochondria are derived from this third-party donor, right? Now, interestingly, because the patient turned out, or the child turned out to be a boy, he will pass none of those mitochondria on to his own offspring. Well, but he also won't be passing on any defective mitochondria because he didn't have those defective mitochondria. Interestingly, on this case, 4 of the 5 eggs that they tried this with died and didn't work. But one of them did and actually went on to do this. All right. So if we're talking about growing humans, how far can we get growing them in a dish? Right? That's everybody's goal, right? Of mad science. We need to have this incubator, you open it up and there's babies growing in it, right? Well, we're actually getting closer to that. We're not there. Don't worry. But some recent studies have succeeded in growing human embryos up to about 2 weeks of age. And that 2 weeks of age is kind of an interesting point because there's been sort of an unsteady rule called the 14-day rule, which says that research should not happen on human embryos older than 14 days. Right? Sort of as a compromise of after 14 days, well, that's maybe too human for us to work on. Anything below 14 days, that's fine because that's just a pile of cells. All right? And part of the reason that scientists agreed largely to this is that up until a few weeks ago, nobody had been able to grow embryos longer than about 7 or 8 days in a dish. After that, you kind of need the trophic support from the uterus and such. But now we're basically right up against that 14-day rule, and so scientists are now asking for some changes to that. And I should say that this rule is not a law. Well, it is in some countries, so I think Britain officially has in their rule books that you can't do anything over this. In the US, it's the National Academy of Sciences highly recommends it, but there's really nothing that could happen to somebody who went beyond that. But people are basically now starting to question how far we can and should be pushing those experiments. and trying to revisit, at least on the national stage and the international stage where there are certain rules in place, questioning whether or not that 14-day rule is appropriate now that we're basically scientifically right up against the border of that. Questions? Thoughts? Yeah?
So, has there been any discussion on the political status of, say, how do you call it, a human-grown ex vivo? So if somehow there was a vat that a human was able to emerge from and be conscious, would they be considered the scientist's property, or would they have their own rights?
Right now, the closest thing that we can say to that has to do with in vitro fertilization. So when a couple does in vitro fertilization, the embryos that are generated are considered their property unless they donate those to science. When they donate them to science, it would, I don't know. You're hitting into some very tight law there, and we'll talk about a few more challenging issues with that in just a second. But the rights of cells that have been donated to science is, I think, still somewhat up in the air. Who has authority over those and what? Probably some of it depends on who you donate it to and what forms you signed when you donate it at this point. But we're not at a point where we are going to be able to produce vats that humans come out of yet. Sure. Right? So I'm just being a little Mary Shelley-ish, right? We're at the early experiments, and we're thinking where are the bioethical frontiers in the next 20, 30 years? Right. And I think you raise a really interesting question that people are going to have to address. All right. Another interesting area right now is in induced pluripotent cells. So you've probably all heard about embryonic stem cells, right? Embryonic stem cells you take from an embryo. For human embryonic stem cells, it generally requires the destruction of the human embryo, which is why it's a somewhat controversial procedure among religious circles. The cool thing about embryonic stem cells is that sort of by their nature, they can make everything in the embryo, right? They have to make all of the cells that produce you, so they're capable of doing all of that, right? And you can actually recapitulate most of that in a dish. If you take those embryonic cells, you can get them to make neurons or gut cells or skin cells or muscle cells or bone cells or heart cells, whatever you want. Right, so people are really interested in this because of the ability to make cells do all kinds of stuff. Right, so that term of being able to make lots of stuff is called pluripotency. Right, the idea of induced pluripotent cells is, what if I could, instead of doing this from just an embryonic cell, what if I could take any one of your cells and do that? Right, and it turns out that we now know how to do that. So we can take an adult skin cell, fibroblasts are just one of the types of your skin cells, by adding a couple of genes to that using a virus. So you basically have a virus infect the cell briefly, give it a little dose of a couple of genes, and then those cells will fix, well, not so much fix, they go back and they sort of reset their DNA to look very much like an embryonic cell, and they start to grow looking very much like an embryonic cell, and then they have the potential to do all of the stuff embryonic cells do. So they can make blood cells, they can make pancreas, they can make neurons, they can make fat cells, they can make muscle cells, they can make anything you want, pretty much. All right? This idea of doing this it's 10 years old this year. It was 2006 was the very first paper showing that you could do this in mice. The paper showing you could do it with human cells came a year later. And where we're at now is trying to figure out a lot of the things that you can do with these cells. All right, so people have now actually made entire organisms from these cells. So you can actually take an egg cell, right, let it develop a tiny bit, and then actually remove all of the cells that would go on to make the embryo and replace them with these induced pluripotent stem cells, right? And you'll actually create an entire mouse out of that, right? So the mouse can be entirely derived from somebody's skin cell at this point. essentially another way of doing like a cloning type process, but via a quite different mechanism, reprogramming first, making these little clusters of cells, then basically sticking that into the developing embryo in a particular way. You can trick it into making a mouse that's entirely derived from those induced stem cells. All right, people are also taking these cells, the ones biasing them to become neurons, and growing them in certain matrices so that they start to look like little human brains. They actually get little folds in them. So brains in a jar. We're getting there on those. They have all kinds of interesting electrical connectivity and actions. Now, I should say these are very small human brains in a dish. Right now, they're about 2 millimeters across. So very, very small human brains in a dish. But that's, as far as modeling diseases, this is a phenomenal bit of research. We don't understand lots of neurodegenerative diseases because we can't really study the animal models. Animal models don't often recapitulate a lot of the human neurodegenerative diseases. So there's hopes that studying neurodegenerative diseases or neural developmental diseases using these little miniature human brains will teach us a lot about how those neurons work, but also starts to make you wonder about what's the thought process going on inside a little human brain in a dish. We can also, in a recent breakthrough, earlier this month, they announced that you can actually turn those induced pluripotent stem cells into oocytes, an egg cell. So you can actually go from a skin cell from anybody and turn that into egg cells in a dish, which you can then fertilize with sperm and make an organism from that. That's great?
No, I said that's crazy.
That's crazy.
How long do skin cells last? Like if I was really creepy and obsessed, and steal some of their skin cells and then
Make
fertilize
one,
them and make
Make your own version of that.
Well, you're actually hitting on a really tough thing there, right? So skin cells, just like what you slough off, are mostly dead ones. But from a little biopsy, you could grow cells for years off of that and keep it alive really easily. You can freeze them down, and then you can keep them for decades.
So, Jason, let me ask you, let's say you have something terrible like your pancreas is failing.
Yep.
Okay, could you take skin cells, do this, and grow a pancreas for this
That's the thought.
And
We're not there
new
yet.
Life by giving them their own pancreas grown from their
Right.
own.
So that's the idea, right? Is that you could now grow hearts or pancreases or lungs or whatever tissue you need to repair, right? That we can't right now. We do some transplants, but the amount of organ tissue is just not nearly sufficient for it. And there's some things that we can't fix, right? We can't do brain transplants, right? So, but there are thoughts if I could give a patient who has a neurodegenerative disease their own brain cells back, right? Maybe we could fix this, especially if you combine it with CRISPR-Cas9. So you can first you can gene edit these cells, right? Fix whatever's wrong, and then you could turn them into whatever you want, right? I think this one is kind of interesting though because it now turns us into a reproductive question, right? One of the things that has always limited cloning research is a limited availability of oocytes, right? Females have a certain number of oocytes. They generally make one a month. You can make them produce a few extras, but it's hard to get human oocytes. This suggests that it could be really easy to get human oocytes, which could totally change what we're thinking about being able to do. All right.
Could I just ask you about...
Yeah.
Could you eliminate the female role in reproduction then?
Probably so. There's probably no reason that you couldn't do this with male cells. I don't think there's any reason that you need 2X chromosomes for the cells to go on to make egg cells. I'm not sure whether there would be a problem in how the Y version of that got itself set. So you may only be able to use the one that makes an X from it, but that would be fine. So taking these cells that can make human neurons, people have stuck those cells into mouse brains, right? This one is actually a little different than that. This one, instead of taking the neurons, your brain's made up of two cells, the neurons that are what everybody thinks do the thinking, and then cells they call glia, which is derived from the same word as glue. People thought it was just the stuff that kind of held the brain together. Turns out those glia are really, really important. In this case, they made a mouse where almost the entire glial set was derived from human glia, not derived from mouse glia. And they did a whole bunch of behavioral tests and showed that that mouse was about 4 times as smart as your average mouse, learned 4 times better, right? So we're quickly heading towards Stuart Little here. There have been a lot of discussions about attempting to create a mouse that has entirely a human brain, meaning its brain would be derived only from human cells. It's not clear what would happen in that case. So right now this is purely a theoretical experiment. They've put a few neurons in, but not derived an entire brain from that. But it's probably just a matter of time before people try that. The people who did this experiment said that they thought about trying it with monkeys, but decided that that was ethically too concerning. And so they were not at this point going to try it with primates, but they were happy doing it with mice. Are humans not that different?
It seems like it raises a question about are all cells created equal?
Pretty much.
In the area of what species are closer to humans than our near ancestors or whatever, primates, what would be a species like pigs, let's say? Do pigs do well?
I mean, so there's a bunch of different. There are trees that show species that are mammals that are closer and further away from us. Bats are kind of relatively close. Mice are not super close. Mice are mostly used because they're so easy to raise. They're just a very common system to do this type of stuff with. All right. So, again, straight from the headlines, you probably have seen this one. This was from September 2016. There's a scientist, a surgeon who wants to do head transplants. All right, and he's preparing to do it next year. They've already identified a Russian patient who has a neuromuscular disease that is almost certainly going to kill him because his muscles can't grow, and basically it's eventually going to cause him to be unable to breathe because you need your diaphragm in order to breathe. But his brain is fine. He's wheelchair bound, and so he has volunteered to become the first of this guy's trials. And the idea is to take somebody who has died recently but whose body is fine and cut the head off and quickly swap the head of this guy who's alive onto that. And then you'd have a functioning head working on this other guy's body.
This is Dr. Frankenstein.
More or less, yeah, we're getting really close to Dr. Frankenstein here. So yeah, there are most people think that this is completely impossible to get to work right now. Nobody, well, I mean, that's what we have to say for all things in science, right? Like, if you had asked 100 years ago, would this be possible? I don't know. So the big challenges here are that your brain starts to die very, very quickly without oxygen, right? And so figuring out how they're going to actually succeed in doing this while keeping the brain alive it's not clear how they're going to be able to do that. Certainly, as of right now, just the act of severing the spinal cord, right, and sticking a new head on, like even if we could assume they can keep the brain alive and like get it hooked back up to the blood vessel so that this other guy's heart that's beating can function, certainly we do not have now any ability to get the neurons to grow from that brain back down the spinal cord. So this guy would almost certainly still be quadriplegic. There's no way to get that information to grow now. Although this guy is working on models of trying to fix that. And so the thing that he has done recently, they've done some work with dogs where they've severed the spinal cord and put in some what do I want to? It's actually a little plastic polymer basically that can trick the cells into growing across it. And one reason to think that this might be possible, I actually got my start in science doing experiments not all that different in goldfish. So we'd go through and we'd crushed the goldfish spinal cord. This fish would not be able to swim. But a month later, they're up and swimming just fine. So they can they're animals, even vertebrate animals, are perfectly capable of repairing their spinal cords. Mammals are not. All right, and we're trying to understand that, but there's nothing theoretical that should stop us from being able to do this type of thing. All right, and so this guy is trying to work on ways of bypassing that wound, basically, and thinks that if he could do that, well, then you could not only do a head transplant, but actually potentially get it to rewire maybe reasonably well and give some even minimal function back to this guy. Whether or not they're going to actually be able to do this next year, I don't know.
So who would that guy be?
Is he a Russian scientist?
No, he's just a guy who's got this. Like I say, he's got this neuromuscular disease that's a degenerative disease. He's wheelchair bound.
I mean, the scientist is going to do
The scientist?
the head transplant.
No, Sergio Canavero. He's an Italian guy. Yeah.
What would the creature at the end? Would
The
it be-
creature?
Would it be like the-
So we've already dismissed this as one of Dr. Frankenstein's creatures. We're not talking about human anymore.
two humans? So what do you call that? It's not really a person. Because their fingerprint, if they got fingerprinted before this, they'd have fingerprints of the body of the guy.
I don't know.
Wow, this sounds like a way to take a false identity.
I think we're developing a cool murder mystery story, though, about the guy who has a head transplant. This is totally a great horror film.
His iPhone won't even work anymore.
You can just do the little reset your fingerprints. It's not that hard.
Do it right before.
Yeah, right. Keep it open. You get that 5-minute window to get the transplant done. It's pretty tricky. Yeah. Yeah.
Get a new iPhone.
So, in your personal,
He can't move his hand, but it's.
so with all this experimentation, in your opinion, where is the moral limit? Would it be perhaps the suffering of the subject?
The suffering of the subject. What do you mean there?
That the scientists wouldn't do something that would inflict physical pain on...
Well, I mean, that turns out to be really tricky, right? Because sometimes you have to do things that cause physical pain to somebody to try to fix them. Like our cancer treatment is more or less, I'm going to try to kill as much of you as possible without totally killing you in the hope that I kill your cancer more than I kill you. It's incredibly painful, debilitating, right? But it's the only thing that we can do. So I don't think that that's actually the limit.
Perhaps long-term physical pain, like crippling someone in a malicious way for, say, experimentation.
I mean, I don't think anybody here is I'm half joking about calling these mad scientists, right? Nobody here is doing this for sort of I want to inflict pain and I'm going to take over the world with my plans, right? They don't have evil layers downstairs and James Bond is strapped to something and their plans get announced, right? All of these things are being proposed for actually very real gains in our understanding of biology. So I don't think any of these scientists or doctors are planning stuff for the purpose of causing pain. But I think you do raise an interesting question about where the ethical line is. And I think actually if there's anything I want you guys to take home from this, it's that we are right at the border, if not well past that in some cases, of where our societal discussions about ethics are. Right? Our societal discussions on ethics are still kind of around the issues about, well, is a fertilized human egg life or not? Science has moved on well past that discussion. Right? We can now create eggs in a dish, we can create sperm, we can do whatever we want. I think that the ethical discussions need to be moving much faster and into the areas where the science is going, because the science is going in some pretty complicated areas. And I think that it's not clear who is actually talking about these in the right ways. Right? So for example, some countries have bans on human cloning. Period. The United States does not have a ban on human cloning. Period. And the reason we don't is because of that distinction between reproductive cloning, actually producing offspring, versus therapeutic cloning. The idea that maybe I could take somebody's cells and then use that therapeutically to create a heart or a pancreas or something like that, right? And the potential from that being a very positive has meant that we don't have a ban on the entire process, right? But then because of that, there's not really any distinction between that. Right, and so I think there's some really challenging things to go there. Mark.
So what I'd like to do is, since we are now officially overtime, let's thank our presenter. But if he's free to stay and you're free to stay, you keep the conversation going until the lights go out. So let's thank you.
Thank you.